students, in the last sessions we have studied the process of biotechnology. There are some process. First, you can say isolation of DNA, then it is cut into fragments, then it is separated by the help of gel electrophoresis. It is that we can say our DNA or recombinant DNA, it is inserted into the plasmid, plasmid is inserted into the host. So that we have studied in the last session, we have studied how to isolate the DNA. It's extracted from the cell by the help of different enzymes. Then we have to cut by the help of restriction enzyme. We will get the small fragments. That fragments we can separate by the help of gel electrophoresis. Then after that we get the fragments and that only we can say it is inserted into the plasmid, that we can say our DNA or recombinant DNA. Now we have to amplify. We have to make different copies, large number of copies. So that process only we can say amplification of DNA. Amplification. The word amplification of DNA. So amplification you can understand making large number of copies. So amplification is done by the help of one of the technique, one of the instruments. It is called PCR. PCR. It means polymerase chain reaction. It is developed by one of the scientists. His name is Carey Mullis. So PCR, it is used to amplifying the particular desired DNA. So there are some steps. First step you can understand denaturation. First step is denaturation. Actually what happens, as we know, DNA is the double stranded structure. So this is the double stranded structure, 5 days, 3 days and 3 days, 5 days. So double stranded structure. So what we have to do, by applying temperature, large amount of temperature, means around 95 to 98 degrees Celsius. A large means high temperature, means we apply high temperature. Because of the temperature what happens, denatured. It means here in between these two strands, as we know, we have studied hydrogen bonds are there. So because of the high temperature, hydrogen bonds will be broken, but two strands will be separated. That process only we can say denaturation. As you know, the word you can understand, denaturation, something breaking. It is denaturing. So by applying high temperature, hydrogen bonds will be broken, two strands will be separated. So two separate strands we will get. Now the second process we can understand, annealing. It is called annealing. So the word you can understand, annealing, it means joining. So there are two strands here. This is one strand and this is another strand. So what we can understand here, temperature is reduced. It means 50 degrees Celsius to 55 degrees Celsius. So somewhat the temperature is reduced. So by the help of less temperature, we can join. We have to join something. That only we can say the process annealing. What we are joining here, a small single stranded DNA or RNA, a small strand, small segment of single stranded DNA or RNA, we have to join here. So a small strand, here also a small strand. So that only we can say primase. It is called primase. So that we have to join in these two segments, we have to join. That process is called annealing. Now the third process, it is called extension. So what happens in the extension, here also we have to increase the temperature like around 70 degrees Celsius to 75 degrees Celsius. We have to increase the temperature again. So what happens in the extension, in the annealing, we have joined a primase, we have joined to start the process. We joined primase. Now we have to use DNA polymerase to extend, to add the nucleotides, to polymerase. We have to use DNA polymerase. But what happens here, DNA polymerase cannot withstand in the high temperature. It cannot withstand. So what happens, we have to extract one of the polymerase. It is called Taq polymerase. So this Taq polymerase, it is extracted from one of the bacteria. It is called Thermus aquaticus. It is extracted from one of the bacteria. It is called Thermus aquaticus. The Taq polymerase is extracted and it can withstand in the high temperature also. So this Taq polymerase, it is used to add the nucleotides. It is used to polymerase. So add the nucleotides. So this process you can say extension. Now, so by the help of this extension, here this the new strand is formed. So at last this two strand becomes four strands. So at last by the help of this PCR we can make billions of copies of DNA. That only we can understand PCR. Now the next process we have to PCR. By the help of this PCR we can make larger copies. Now the rDNA we have to isolate. We have to select by the help of selectable marker. Now the small process, last process it is that we can study in the next video. Please listen the next last video of this chapter. Thank you.